Interphase. Similar to mitosis, meiosis starts with an interphase, during which a cell's chromosomes duplicate. Once finished, each chromosome consists of two genetically matching sister chromatids, which are attached to each other by a centromere. Prophase 1. Early in this phase, the chromatin coils up. Homologous chromosomes are made of two sister chromatids, and these chromosomes come together as pairs. This results in a tetrad. The chromosomes then exchange segments by a process called crossing over. As prophase continues, the chromatids condense more as the nucleoli disappear. The centrosomes then move away from one another and a spindle begins to form. Metaphase 1. In this stage of meiosis, tetrads line up on the metaphase plate halfway between the two poles of the spindle. The sister chromatids of the chromosomes are still attached at the centromeres, and spindle microtubules are attached to kinetochores at the centromeres. In each tetrad, sites of crossing over hold together homologous chromosomes. Also, in a tetrad, the spindle microtubules attached to one homologous chromosome comes from one pole, while the spindle microtubule attached to the other homologous chromosome comes from the opposite pole. Therefore, the homologous chromosomes of each tetrad are positioned to move towards opposite poles. Anaphase. During the anaphase in meiosis, the homologous chromosomes or tetrads separate and move to each pole of the cell. This is similar to anaphase in mitosis, however, the difference is that during meiosis, the sister chromatids stay attached at their centromere. So, 3 still doubled chromosomes then move toward each spindle pole. Telophase and cytokinesis. During telophase 1, the chromosomes arrive at the poles. Each pole of the cell has a haploid chromosome set. At this point, each chromosome is still made up of 2 sister chromatids. During cytokinesis, the chromosomes begin to uncoil. Also, the nuclear envelope begins to reform. The first meiotic division begins the preparation for the second meiotic division. There is no chromosome duplication between telophase 1 and meiosis 2. Meiosis 1, the process starts again. The difference between meiosis 1 and meiosis 2 is that in meiosis 2, the process starts out with a haploid cell. The chromosomes begin to condense, and the outside nuclear envelope begins to break down. During prophase 2, a spindle forms, and the chromosomes begin to move towards the center of the cell. In metaphase 2, the chromosomes are lined up on the metaphase plate. The sister chromosomes separate into daughter chromosomes in anaphase 2. The daughter chromosomes then move towards opposite poles in each cell. During telophase 2, the nuclei begin to form at each pole, and the nuclear envelope begins to reform. At the same time, cytokinesis occurs. Finally, there are 4 daughter cells, each with a single number of chromosomes.